So there are different strategies that one can use to get into a state of nutritional therapeutic ketosis. There is starvation, that's not fun, but there's caloric restriction, there's intermittent fasting, there's the ketogenic diet. So now we know, I'm just coming from an epilepsy conference, that the modified ketogenic diet, which is what you see here, or at least the ratio is about 75% fat, 20% protein, and a minimal amount of carbohydrates, like 5%, seems to have similar seizure protection in adults and maybe in some pediatric populations too, depending on the disorder. So these things, basically starvation and the ketogenic diet and intermittent fasting, mobilize fats. The fats go to the liver and it stimulates beta oxidation of fats with the continuous suppression of insulin. So only in the context of mildly depleted liver glycogen and continual suppression of the hormone insulin, and blood glucose drops a little bit too, but the homeostatic mechanisms that maintain blood glucose are very powerful. So blood glucose really does not bottom out. It might stay in the 70 to 80 milligram per deciliter range, even with no carbohydrate consumption for many people. So these ketone bodies are produced, beta-hydroxybutyrate, acetoacetate. Acetoacetate can spontaneously decarboxylate to acetone. Interestingly, acetone has some interesting functions on various ion channels, which can affect brain function. And this is difficult for patients to achieve. So we developed various forms of exogenous ketones and also acknowledged that fats, ketogenic fats like medium chain triglycerides, which go directly to the liver via hepatic portal circulation and are oxidized very fast to ketone bodies. They're not packaged into chylomicrons like the long chain fats. And then ketone salts and ketone esters are metabolized partially in the liver, but mostly spill into the blood and then rapidly elevate ketone levels. Like a ketone ester, if a person consumed it right now on stage, by the time I get off stage, it'll look like I fasted for 10 days. So you can achieve a level of ketosis in the 5 to 6 millimolar range, go from 0 to 5 to 6 within 30 minutes. So they are very powerful agents that we're studying. But as the science sort of advanced, we understand these molecules not only as energetic substrates for the brain, the heart, and even skeletal muscle, but they have powerful signaling properties in the brain and in other tissues. So I'm not going to go through all the signaling pathways here, but you can see in this panel below that there are many different signaling pathways that are impacted by nutritional ketosis.
ROS, reactive oxygen species, also called oxygen-free radicals, are a side product of sites on mitochondrial complexes of the electron transmitter chain. They can contribute to membrane damage by lipid, fat, peroxide formation, leading to apoptosis, programmed cell death, including DNA fragmentation and mRNA decay. TCA cycle, citric acid cycle, or the Krebs cycle, a series of chemical reactions to release stored energy through the oxidation of acetyl-CoA, derived from carbs, fats, and proteins. GABA, gamma-aminobutyric acid, an amino acid that acts as an inhibitory neurotransmitter, blocking messages sent between the nerve cells and the brain or spinal column, thus reduces fear, anxiety, and stress. Glutamate, a non-essential amino acid, the anion of glutamic acid as a neurotransmitter, the most abundant excitatory neurotransmitter in the vertebrate nervous system, accounts for over 90% of synaptic connections in the brain. Has to be present in the right places, right time. Too much or too little is harmful. Adenosine, one of the 4 nucleoside building blocks to DNA and RNA. Derivatives include the energy carriers adenosine mono, di, and tri-phosphate, AMP, ADP, ATP. GPR109A, hydroxycarboxylic acid receptor 2, niacin receptor 1, a protein encoded by receptors that are primarily stimulated by beta-hydroxybutyrate, which metabolizes into acetoacetate, the first ketone body produced in a fasting state. NLRP3 inhibitors. These inhibit NLRP3 inflammasomes. Their beneficial effects in several diseases, including Alzheimer's. They also help enhance longevity.
So changing our metabolic physiology significantly changes brain energy metabolism and also the neuropharmacology of the brain. So one of the studies that we did in Angelman syndrome, we take brain tissue out and we look at all the different neurotransmitters, and the ratio of glutamate to GABA changes significantly. It activates glutamic acid decarboxylase, which makes an excitatory amino acid transporter, an inhibitory or brain stabilizing neurotransmitter known as GABA. So GABA also has anti-seizure effects. So it's one of a dozen different mechanisms, and that's what's really fascinating about the ketogenic diet. It doesn't just work through one mechanism. It works through many different mechanisms in synergy. And that's why there are no drugs that really can mimic the ketogenic diet, because it works through so many different mechanisms.
Stated and summarized, easy to share with loved ones. The description below the title for this video has these summary points.